All right, hello. Today we're joined by Dr. Asma Zaidi from the KCU Kansas City campus. Welcome, Dr. Zaidi.
Thank you very much, Dr. Creamer. It's great to be here this morning.
Well, today we're going to be talking a little bit about the research that you're doing in your laboratory. Um, why don't we just start off with kind of a 30-second elevator pitch of your current research focus?
I love that. Thank you so much for this opportunity. So, I work on brain aging and neurodegenerative disorders such as Parkinson's disease and Alzheimer's disease. And we are trying to address three major questions. So, number one, what are the biochemical changes that occur in neurons during brain aging that lead to cognitive dysfunction in the elderly? Aging itself is really not a disease per se. It's a perfectly normal physiological condition, but it is the biggest risk factor for developing neurodegenerative disorders such as Alzheimer's and Parkinson's disease. And, these are very severe pathologies, right? So clearly something happens in the aging brain that predisposes one to develop these neurodegenerative disorders. And we're trying to kind of figure that out. What is the bridge between the two? And number two, one of the hallmarks of neurodegenerative disorders is the selective vulnerability of neurons. So it's always like a group of cells or a population of neurons seems to be dying, whereas the rest of the brain is healthy. For example, in Alzheimer's disease, the hippocampal neurons are degenerating. In Parkinson's disease, it's the dopaminergic neurons in the substantia nigra that seem to be targeted. In Huntington's disease, it's the cells of the basal ganglia. And so to date, we do not understand why these specific groups of neurons are targeted, whereas the rest of the brain seems to be pretty healthy. So we are trying to address and tackle this question as to this unique vulnerability of selective populations of neurons. And number three, obviously, we want to be able to prevent these, changes, these negative impairments, or reverse them if possible. And to address this, we are using and testing a variety of different compounds, such as flavonoids, dietary flavonoids, which are pigments present in fresh fruits and vegetables, also healthy oils, so omega-3 and omega-6 fatty acids, and testing their efficacy in neuroprotection. So those are kind of the three areas that we are pursuing in my lab currently.
Great. That's really interesting, especially to me the dietary components. Are you looking at that? And I don't know the research in this field very well, but are you and others looking at these dietary components in terms of whole foods or in terms of supplements or maybe both? Both?
So we are using cellular models to
Okay.
test their efficacy. So our experimental paradigm is as follows. So we, culture neurons, and we expose them to different neurotoxins that simulate many of the biochemical characteristics of, let's say, Parkinson's disease. So the compound that we use is 6-hydroxydopamine, and it induces a lot of those changes that occur in humans with Parkinson's disease. And so we pre-treat these cells with compounds. So these are purified compounds, the flavonoids. So we've been testing, curcumin, resveratrol, EGCG, which is present in green tea, another compound called fisetin, which is present in strawberries, and then testing the cells for increased viability, because the neurotoxin kills the cells. So we see if these compounds can prevent cell death. And then going into a more molecular level, so there is a lot of literature, much of it comes from my lab, showing that there is a disruption in calcium homeostasis in aging as well as neurodegeneration. And the defect seems to lie in a protein called the calcium ATPase. So this is a protein that I've been working for many, many years. It was the topic of my PhD thesis, as well as several postdoctoral positions. So this protein, and we were the first ones to show that the calcium pump is damaged in the aging brain and in Parkinson's disease. So, we do kind of the general viability assays, but we also do more specific mechanistic type types of assays to see, if the flavonoids protect this calcium pump from the neurotoxin, or if intracellular calcium, so when the pump is damaged, intracellular calcium is elevated, and this high calcium then activates calpains and caspases and the cell death pathway. So we are able to measure in live cells the amount of intracellular calcium. And so, once we've done the treatment, we see if those calcium levels are reduced by the treatment. We
Excellent.
also look at the formation of reactive oxygen species. So when you expose cells to neurotoxins, there's a lot of production of free radicals. So that's another measure that we use to test the efficacy of these compounds. Levels of glutathione, of superoxide dismutase, so antioxidants. So it's a whole array of
Hmm.
different, parameters that we test when we're measuring the efficacy of these compounds.
That's interesting. Kind of along the same lines, and again, this is just due to my inexperience in the field. Uh, have you looked into, or are people looking into the idea of dietary components that maybe have a negative effect on this neurodegeneration, or is it more of a protective where the field is at?
So the literature shows a protective effect. I mean, there's in the last 2 decades, I would say there's been a lot of work done on dietary flavonoids and also like epidemiological studies. For example, in India, where people use curcumin in their food every day, there is less incidence of Alzheimer's disease. We call it the French paradox, So in France, people drink a lot of red wine, which contains resveratrol, and that has been shown to have some cardioprotective effects. So I think to answer your question, it really is dose dependent. Excess of anything can be bad. Anything that's an antioxidant can become a pro-oxidant if it's taken in larger amounts. So, of course, there needs to be a nice window of the dose that one needs to take. And it's very complex. As human beings, there's going to be differences in consumption, or, the type of food that you're eating, or what time of the day you're eating, how much you're eating, all of that comes into play. Your genes obviously play a role itself. But in my research, we are kind of approaching it from a very simple cellular level and really addressing the question of whether these flavonoids can protect this calcium pump, the target that has been shown to be damaged in aging and in neurodegeneration.
All right. Well, my next question for you, Dr. Zaidi, is how do you feel that working in your lab will help prepare a medical student for a career in medicine?
Yeah, that's a great question. So I'm going to tell you that I'm very hands-on with my students. I communicate with my students the enthusiasm and the passion for learning, for being curious, for kind of being abreast with what is being discovered, right? Because, the field of medicine and life sciences is so dynamic. There are new discoveries being made. So just the students, I actually, tell them to stay in touch with where the field is going and to be engaged in lifelong learning, right? So my philosophy is that, one can be a successful physician if you know your diseases and if you know your treatments, right? You treat the patient and they heal, and you are a successful physician. But I think that you will be a superior physician if you knew what was happening in the disease, like what is causing the disease and how are these pharmaceutics working. If you knew those two things, you would be a far superior doctor and help your patient a lot more than just being able to prescribe medicine for their ailments. So kind of, being a part of research as you are a medical student, and hopefully, one day when you are a physician, just keeping abreast of what's happening is very important in my opinion.
Yeah, we're seeing that currently with our
Hmm.
pandemic and
Yes.
learning the importance of just basic science and basic research
Hmm.
in dealing with a health crisis. It's very
Absolutely.
important.
Absolutely.
So, your time at KCU, approximately how many students or residents or fellows have you worked with or trained? And what are some things that they've gone on to do after they've worked with you?
Yeah, so I have trained several dozen students. I've been here at KCU for 12 years now, and I've had the unique fortune of being able to train, many students. I remember my very first student. She is now a plastic surgeon in the New York area. Several other of my trainees, they are in medical school. Some of them have graduated and gone on to, successful careers in medicine. Yet others are actually, there were several students who went on to PhD programs, and a few have graduated. One of my students graduated from his PhD and now works at a big pharma company, Amgen. Another one of my student, Paul Ramlaw, who did a 2-year research track thesis with me, is the biosafety officer at Children's Mercy Hospital. Another student is in the DC area and overseeing clinical trials. So, really, there's a whole array of different career options that the students have taken, which is wonderful because normally we sort of limit ourselves to either being a researcher or being a doctor, but there's like so much that one can do when you have the right training and the right passion and the right questions.
Yeah, excellent. It sounds like you've done quite a bit with students. So, on that note, if you could give one piece of advice to medical students currently, what would that be?
Yes. So my advice to medical students is work hard, be disciplined in your learning. There's really no substitute for that. We all know that. But having said that, keep that passion for learning, have that curiosity of how things work. And, I know med school is very difficult. So when things get difficult, just remember the reason why you wanted to be a doctor, the values, your ideals, and the reasons that brought you to medical school in the first place. Just remember that. um, be kind and compassionate to one another, to your peers. People may be going through difficulties. Be tolerant and accepting of all types of people, regardless of their ethnicity, color, creed, religion. But above all, love yourself and take care of yourself, and find time to do things that you enjoy. I think replenishing your cup is so important. So that's what I have to say to my students in the com.
That's excellent, Dr. Zaidi. I know our students appreciate you very, very much, and they can see your enthusiasm no matter how they're interacting with you. And so we really appreciate you taking some time today to talk about the research that you're doing, and some of the opportunities that students would have working with you in your lab.
Thank you very much, Dr. Kramer, for this opportunity to share about my research. And I want to thank my students for being passionate about learning. And, it takes two to tango, and it's a two-way street, right? So if they are not receptive of the passion, my passion will hit nowadays a computer screen, right? But I feel like it goes beyond the computer screen and goes into the hearts and minds of the students on the other side. So I'm very appreciative, and I'm very fortunate to be able to work with such, wonderful students at KCU.
All right. Well, thank you very much,
You
and
bet. Thank
we'll be
you.
seeing you around campus or on
All
Zoom.
right. Yes. All right. Thank you.
Bye
Take
bye.
care. Bye.